All right, let's talk about the last part of the central dogma. That is taking the messenger RNA we made in transcription and translating it into a protein. Translation occurs in the cytoplasm. So the first thing that has to happen is that mature messenger RNA has to be exported to the cytoplasm. Once in the cytoplasm, a ribosome complex will form around the messenger RNA. The cap of the RNA will be in the P site of the ribosome, while the first codon will be in the A site of the ribosome, and this will begin the process of translation. Once the complex is formed, the first transfer RNA, which will have an anticodon matching the first codon of the messenger RNA and an attached amino acid, will come in and bind to the A site. In this case, it is methionine. With the transfer RNA in the A site, the ribosome will then move down one position, one codon, leaving the A site open and putting the first transfer RNA in the P site and the cap in the E site. With the first transfer RNA moved over to the P site, the A site is open, and a new transfer RNA with an anticodon for the codon in the A site comes in and binds. At the same time, a bond is formed between the two amino acids that are now adjacent. In the next step, we have the original transfer RNA being released. The second transfer RNA, now with two amino acids attached to it, moving to the P site, and the A site opened up again for another transfer RNA to come in. When the ribosome moves down and the A site is free, a transfer RNA with a correct anticodon comes in, carrying an amino acid. That amino acid is joined to the others in the growing chain, and the process will continue. This entire process of moving down the messenger RNA, adding a new transfer RNA, releasing the old ones, and growing the chain together will continue until you reach a stop codon or a pause in the process. When the ribosome reaches the end of the transcript or hits a pause, the entire mechanism stops and the process comes to a halt. When the process stops, the players separate from one another. We have a ribosome that can be reused, find another messenger RNA and make another protein. We have a messenger RNA that can go find another ribosome and make another protein. And we have the protein that we were trying to make, the long chain of amino acids. The transfer RNAs are in the cell and they need to have new amino acids attached to them, but they can also be used to build new proteins. This sums up the translation of DNA into RNA into a protein that will carry out a job for the cell.